my name is Malia Tapia, and I will be presenting my portion on our group project, which is on the Asian blue tick. To give a description of this, they are a hard-body tick species. Then the size for a female or like typical ticks are 3 to 5 mm long, but for females, they can get up to 12 mm long when they are fed. They do have a blue-gray hue to them, but they are reddish-brown when they're unfed. Their body shape is it's about like a teardrop shape, and their mouths are short. And they do have a hard shield for a back, and the females have smaller ones just to let the body expand when they're feeding. For hosts and environments, they are one host tick, so this means that they only feed on one single host for their entire life stages. The primary host is cattle, but there are also other hosts, including livestock like horses, sheep, goats, and also deers. But there are also rare occasions of them biting humans. And for the environment, they do strive in warm and humid climates, and they are typically seen in grasslands. For the disease, the bovine babesiosis, which is also known as cattle fever, the pathogen responsible is the Babesia bovis and the Babesia bigmina, and the type of pathogen is single-celled parasites. And the associated symptoms with this cattle fever is high fevers, anemia, weakness, and loss of appetite. And the diagnosis is in 4 stages, which is clinical signs, examination, serological examinations, and molecular testing, which is all done through the veterinary. For treatment and prevention, there are anti-protozoal drugs, there's anti-inflammatory drugs, blood transfusions, and fluid therapy. And for the prevention of this disease, there is the vaccinations against the Babesiosis bovis and the Babesia bigmina, and there's also just managing these grasslands is another prevention method. And then also there are acaricides that can be used for prevention.
Hi, my name is Panya Gadekar, and I'll be going over part 2, the epidemiology and control measures of R. microplus. The most recent outbreaks, considering the last 10 years, were in Cameroon, Africa, in Uganda, and South and Central America, which had the most drastic ones. In Cameroon, Africa, there were many reports showing the displacement of the native tick species and the increased population of R. microplus. And this is kind of concerning because this tick is invasive, resistant to acaricides, and is a vector of several livestock diseases. In Uganda, there was a study showing the same results and showing that R. microplus was dominating the region, whereas the native ticks were not there. In South and Central America, there's so many consistent outbreaks that the disease has become endemic. The prevalence nationally was in Texas, regarding United States, specifically southern Texas, including counties such as Cameron, Starr, Willacy, Val, Verde, Webb, and Jim Hogg, showing and encompassing infected quarantine premises. This is primarily due to the border with Mexico. Internationally, regions of the world that are affected by this disease are tropical and subtropical regions. It's endemic in Mexico, many parts of Central and South America, parts of Asia including India and Pakistan, and there's an emerging spread in Africa, specifically Burundi and Cameroon. The cattle fever tick, or Rhipimphalus microplus, vectors, lethal bovine diseases, and functions for motor tick neuropeptides are unknown. But in this study, the researchers go through using RNA interface, go through RumiCap2BR, and it's confirmed that the expression of the receptor resulted in major physiological disruptions. Basically, the females were weakened, and overall, the results indicated that blocking the signal pathway might be an effective strategy for tick control and has positioned the signaling system as a promising target for new anti-parasitic interventions. So this is a really interesting thing to look forward to in the future, and this might significantly help lower the number of the Asian ticks, not only for the environment, but for the public health and for the health and safety of livestock. Thank you.
We're going to talk about the impact of the Asian blue tick disease. So when it comes to public health, the Asian tick affects the public health directly and indirectly. Ticks, they pose a direct and indirect health risk. Directly, ticks can carry pathogens like the ones we've been talking about in the past slides, like the Babesia bigmina, and that affects cattle, putting cattle's handlers and farmers and the veteran workers at risk of tick bites and allergic reactions. Indirectly, heavy infestations of the Asian blue tick can lead to like increased use of like the tick killing chemicals that kills the ticks, hopefully to like help the cattle, which can harm the environment and also do real damage to the health of others, especially other organisms, not really to humans, but but I will say that the ticks do sometimes carry pathogens that can be harmful to humans, but majority of the time it's only harmful to the cattle. Yeah, and additionally, the tick-borne disease can the disease like reduces like the meat and the milk production, and impacting like food and the agricultural. Next, we are going to talk about the economy. So livestock, the cattle, they're very important for the economy. So when the Asian ticks impact the cattle, it reduces like the milk yields. There's slower growth, decreases fertilization, and like there's lower meat quality. So this all like cuts into the profits that farmers are making, and that starts impacting the economy. Additionally, like the risk of tick spread can result in trade restrictions. So because they want to limit the spread of the ticks, countries may have like different restrictions on what can be exported. So the reducing of the export of like the beef and the dairy products can have a really bad impact on the economy, and this can also end with billions in global losses. And then farmers, they have to spend heavily on care for the cattle and like the tick killing chemicals. So that also has a huge impact on the economy. Last, we are going to talk about the environment. So first, the chemicals that are used to kill the ticks can sometimes run off into the soil and water nearby, causing pollution and affecting the ecosystem. Not only do they like harm the ticks, but they also have a negative impact on the wildlife and the insects that are nearby, because they're not the ones that are being targeted, but they are the ones near and get impacted. What's more is that the Asian blue tick is rapidly developing resistance to the chemicals. So this is like forcing the farmers to increase their dosage and trying to control the Asian tick infestation. To make matters a little bit worse, because of climate change and the warmer temperatures increase, ticks they thrive in warm temperatures. So this allows them to spread into new habitats, further like spreading the issue. So this growing cycle of chemical use, resistance, and environmental damage creates a significant challenge for both the agriculture and the environment.
Hi, my name is Riley Murdoch, and I will be covering what the research tells us. Also, the research article that we've chosen is Tick Saliva-Induced Program Death 1 and PD Ligand 1 and its related host immunosuppression.
Rhipicephalus microplus RM is a one-host tick that feeds on cattle. The tick causes many negative effects on cattle, including loss of blood, slowed weight gain, and reduced milk production, and transmit diseases such as Babesia species and Anaplasma species to cattle.
Acaricides, which are pesticides for ticks, are used to control these ticks, but they have many adverse side effects. This includes the selection of resistant tick populations to these chemicals, as well as food and environmental contaminations. The development of an alternate control strategy, such as tick vaccines, are needed, and to do this, we must understand the tick. Tick saliva contains a few immunosuppressants. This includes cytokines, serpins, metalloproteinases, lipocalins, PGE2. PGE2, or prostaglandin E2, is an inflammatory mediator. It induces programmed cell death, PD1 and PD ligand 1 expression in several animals, including cattle, and it has been found in other tick species' saliva. Since PGE2 is an inflammatory mediator, it regulates the activity of immune cells such as T cells, dendritic cells or DCs, macrophages, and it contributes to immune evasion during cancer and chronic infection. PGE2 inhibits the Th1 immune responses, including T cell proliferation and Th1 cytokine production in cattle. So the results and discussion. This image is from the research article showing PGE2 and PDL1 expression in R microplus in attached versus non-attached sites. The C and D sections at the bottom specifically show counts of immune-positive cells in the sample areas. This study demonstrates the existence of PGE2 in RM saliva. The PGE2 concentration in RM saliva is 200 nanograms per milliliter, which was much higher than that in the plasma from healthy cattle, which is 73 picograms per milliliter, which is approximately 2,740-fold greater. This shows immunosuppressive effects of T cells, macrophages, and dendritic cells. The RM saliva is related to PD-1 and PD-L1 upregulation in host immune cells. They state that it is unclear whether PGE2 in tick saliva is involved in the upregulation of other immune cells and molecules, and they call for additional experiments to address this. Thank you for listening, and here are all of our sources.